What is the first thing you think of when you hear the words stem cells?
Saving a life.
Politics.
Little babies, unhatched babies.
Future. Abortion, I suppose.
I think research for they use that for research for what, spinal injuries and stuff. I think that what Christopher Reeves was. Yeah.
Research.
Why?
Because there's so much talk about it. Pros and cons.
The controversy between how much we should use stem cell for and how much we shouldn't, and what's playing God and what's not.
From aborted fetuses, then I have a problem with that because I'm a Christian, and then I also have a girlfriend who's all into this stuff. I see the good it can do, but I'm just not sure where I stand on is a fetus or taking stem cells from a baby, or is that a bad thing versus what good it would do? I don't know.
Babies.
Okay. Where does that come from?
TV, just in general, TV, the abortion thing, the whole issue about that, the research that they're doing, a lot of the political can't do this kind of thing. Personally, I think it's fine. That's just me. I think stem cell research is an important part. We're humans, we have to advance, we have to have a backup plan.
Well, as you can see, I'm pregnant, but before this pregnancy, I actually had triplets, and I did in vitro fertilization, and I actually had 3 leftover eggs, and I had to decide what to do with them. And one of the options was donating them to science for stem cell research. And it was a tough decision, but that's what we decided to do. So it's a topic that's close to my heart.
In any polarizing debate, you typically have a distribution of opinions that looks something like this. On one end, you have the people that are strongly against it. And on the other end, you have the people that are strongly for it. And the people on the ends are by far the most vocal within the debate. Unfortunately, they're also the ones that are most oftentimes the source of the misinformation. And the same holds true for the debate about stem cell research. It would be nice, wouldn't it, to clear up some of the misunderstanding and misinformation that revolves around this debate? But how can we do that? We live in the information age. We have TV, 500 channels. We have the internet. You'd think it'd be easy, but it's not. See, most of what we read or hear has a spin to it. It's those two ends trying to get you to agree with their side of the argument. It's my hope to provide you with a factual understanding of stem cell research. You see, I'm okay with whatever side of the fence you want to stand on. I just want you to base your decision on the facts. So where do we begin?
The good sisters of St. Francis taught me that the best way to understand something is to read about its history. For centuries, science has been making discoveries that shake up our worldview and force us to think about our lines in the sand.
The stem cell debate began with this, a microscope. And this tool has its origins some 1,200 years ago. You see, for the very first time, we could peer into the inner workings of the world around us. And given our inquisitive human nature, that's just what we did. It took some time for us to get going, but we eventually turned the lens inward and began the discovery of what makes living things work. In 1665, Englishman Robert Hooke coined the term cell, describing what he saw while examining a piece of cork. It wasn't until the early 1900s that scientists in Europe finally discovered the cells in our bodies, which produce our blood cells. This marked the first stem cell discovery. But it wasn't until the late 60s that the research on stem cells really took off. In 1968, the first bone marrow transplant was performed. In 1978, stem cell lines were discovered in human cord blood. In 1981, the first in vitro stem cell line was developed from mice. In 1988, embryonic stem cell lines were created from a hamster. In 1995, the first embryonic stem cell line was derived from a primate. And in 1998, Dr. James Thomson from the University of Wisconsin-Madison isolated cells from the inner cell mass of early human embryos and developed the first embryonic stem cell line.
So now that we know the history, we need to find the facts. Where do these embryonic stem cells come from? What are stem cells? What can they do? How do they do it? What is the purpose of this research? In other words, how will this research eventually help us? To answer these questions, I thought that the best place to get the facts about stem cells was to talk to the man who produced the first embryonic stem cell lines, Dr. James Thomson.
So your body's made up of different kinds of cells. And some cells look like brain cells, and some cells look like heart cells, and they differ because they express different kinds of genes. And when you damage some of the cells in your adult body, like if you cut your skin, it repairs itself. And part of that repair process is through something called a stem cell. So the job of a stem cell is to replace itself so it doesn't go away with time, and to form all the parts of that particular tissue it lives in. So in the case of skin, it forms the layers of your skin. There's stem cells in the blood, whose job it is to make all your blood cells. There's stem cells in your gut, it replaces your gut cells. And in the adult, these stem cells give rise to just a few other things. Sometimes it's a fairly large number of things, but usually it's one or two different kinds of cells.
What do stem cells do in our body?
So there's different kind of stem cells. In our adult body, stem cells are, for example, in your skin. And when your skin turns over, or if you have a sunburn and some skin comes off, the cells that replace that are called stem cells. So their job is to replace themselves and then differentiate to whatever makes up the tissue. So at the base of your skin, there's a stem cell, but it only makes skin. And you have stem cells in your blood that make blood and other places in your body. But in general, these adult cells can only give rise to a limited number of things. So they're kind of stuck in their fate.
What are embryonic stem cells?
If you go back really early in development, there's cells that aren't restricted to what they can make, and they can form everything in the whole body. And these cells, these cell lines derived are called embryonic stem cells, and they can replace themselves by proliferating in this undifferentiated state forever, and yet they have this ability to form everything in your whole body. So that means that we can, for the first time, make very large numbers of cells that make up the human body.
Why don't we use the adult stem cells and avoid the controversy altogether?
And most cells don't have those properties. Most cells, if you put them in tissue culture, they divide a set number of times and die. There's this very limit, it's 60 to 70 doubling, something like that. Whereas these early embryonic stem cells have the genetic machinery to divide forever, and they're normal. And that means you can make as many cells if you want of particular parts of the body without limitations.
Where do embryonic stem cells come from?
The ones that we used were from IVF clinics, and that's in vitro fertilization. Couples that have infertility problems elect to have this procedure. Most people, if there's more embryos than they can actually have implanted, choose to have them frozen. And that means that they can use them at a later date to attempt to have a child. And sometimes, for whatever reason, often the couples have had the number of children they want, and they still have these frozen embryos. And they have to come to a decision of what to do with them at that point.
This is where it happens. Couples come here to get help with having a baby. For whatever reason, they can't do it alone. They need the scientists and the doctors in here. And since this is the source of the embryonic stem cells, it's important that we understand how this process works. So why don't we take a look at both the natural process and the in vitro fertilization process.
In the natural process, once a month a woman's ovaries release an egg into the fallopian tube. For the next few days, the egg continues its journey toward the uterus. If during this time sperm are present, the egg can be fertilized. Once the egg has been fertilized, it is now called a zygote. During a week-long journey along the fallopian tube, the zygote continues to develop until it resembles a fluid-filled ball of cells with a small raspberry-like cluster of embryonic stem cells floating inside. It is now called a blastocyst. After about 7 days, the blastocyst reaches the woman's uterus. If the conditions are right, the blastocyst will attach itself to the wall of the uterus. Once the blastocyst is firmly secured to the uterine wall, the remaining embryonic stem cells begin their division into the specialized cells that make up a human baby. And if all goes well, 9 months later, a baby is born. In the in vitro fertilization process, a woman is given daily injections for about 10 days to stimulate egg production in her ovaries. Doctors then remove the eggs from the woman. Once the eggs have been checked for developmental potential, they are mixed with sperm from the husband in a petri dish. If necessary, a manual implantation of the sperm can be done by an embryologist under a microscope. The now fertilized eggs are incubated in a heated cabinet for 2 to 5 days. While in the incubator, the fertilized egg, or zygote, divides much in the same way as it does while on its journey down the fallopian tube. When the developing zygotes have reached the blastocyst stage, they are ready for implantation back into the woman's uterus. They are then removed from the incubator and are graded. The doctors choose the best candidates, usually 2 to 4 of them, and then freeze the remaining ones for future use. The doctors then implant the blastocysts into the woman. If one or more of the blastocysts have adhered to the uterine wall, then development of the embryo will occur and follow the natural course of development. If, however, none of the implanted blastocysts have attached, the doctors can then thaw some of the remaining blastocysts and repeat the procedure.
Now that we understand both the natural process and the IVF process, there are a couple things you should know. First, not every fertilized egg or zygote becomes a baby. In fact, about 18% of the time, it doesn't happen. Call it fate, chance, or God's will. It just fails to become a baby. And for some couples, it never happens, which is why the IVF procedure is necessary. The second thing you need to understand is that not every blastocyst implanted via the IVF process becomes a baby. Again, call it fate, chance, or God's will. It just doesn't happen. In rare cases, all of the blastocysts implanted adhere, and the couple then has twins, triplets, or quadruplets. The third thing I need to share with you is the fact that as a result of the IVF process, there are excess blastocysts. The couple having the procedure is oftentimes left with a very difficult decision: what to do with the remaining frozen blastocysts. In fact, they are left with only 3 alternatives. 1, they can donate them to other couples. 2, they can donate them to science for stem cell research. Or 3, they can dispose of them down the drain. You see, the blastocysts cannot be frozen forever. Freezing them really just delays the decision. So the question remains, what to do with these extra blastocysts? Yes, they have the potential to become a life, but there are no guarantees that that will happen. Just as we have seen in both the natural process and the IVF process, both do not always result in a pregnancy. Can you call this abortion? By definition, abortion is the termination of an ongoing pregnancy. These unused blastocysts have the potential for life, but are not yet securely growing inside a woman's womb. So we can't confuse this with abortion. This is the point where we each have a decision to make. People have very different opinions about whether these blastocysts are life or not yet life. And that's okay. We live in a democracy. It's part of the foundation of who we are as a country. We agree that it's okay to disagree. So each of us has to decide for ourselves what is the right thing to do with these extra blastocysts. This is about our own individual line in the sand.
There's more controversy regarding an implied link to cloning. Is cloning related to stem cell research?
Yeah, it's majorly linked in the public's imagination because Dolly was cloned in 1997 and human embryonic stem cells were derived in 1998. And in Dolly, a nucleus from a differentiated cell was taken and put in an egg. And then that nuclear transfer product was put in a sheep and it formed Dolly. And what people thought was you could do the same thing with human embryos or human oocytes, and instead of actually implanting it back into a uterus, to use it to make an embryonic stem cell line. And that embryonic stem cell line would be genetically matched to the donor of the cell, the fibroblast or whatever. And the idea was that this would be a perfect match, and if you used derivatives of that cell line, it wouldn't be rejected by the immune system. So it'd be your own personalized cell line. And there's no biological reason why that can't work, but it's always been my feeling that it would be so inefficient. I think it's economically not viable. You just need so many oocytes for a very inefficient process that, given the number of people with diseases that are, potentially treated by embryonic stem cells, would simply never happen. So I always thought that technology would just go around it, and we'd find other ways to reprogram cells that don't actually require nuclear transfer in oocytes.
We now know what stem cells are. We know that stem cell research is not related to cloning. We know that stem cell research is not related to abortion. But what we don't know yet is the how and the why. So let's find out, how do scientists reprogram stem cells into the other cells that make up our body?
They're grown in these little tissue culture flasks, and there's a lot of different ways of growing them. But they have a fluid medium in the top, which is pink. And you put different chemicals in that medium that act on the cells to direct them in different ways. So depending on the lineage, scientists are already good at making some cells like brain cells and not so good at some other cells, but I think there's going to be tremendous progress over the next decade or so.
Dr. Thomson and Dr. Shinya Yamanaka figured out how to manipulate a living skin cell from an adult and tell it to revert back to its original embryonic stem cell state. They could then take this induced pluripotent stem cell and grow it into whatever they needed it to be. A heart cell, a bone cell, a nerve cell, actually any of the over 200 different cells in our body.
There were several ideas for human embryonic stem cells, but the newest idea is to use these induced pluripotent cells, which, as far as we can see, are equivalent to embryonic stem cells. And these are cells where you can take a fibroblast from your skin, add 4 genes, the different genes by different groups, but it converts those cells back to something that's developmental equivalent to embryonic stem cells as far as we can tell. And those cell lines would be completely matched with the individuals. Your cells that are like liver cells or heart cells or neural cells all look different, and they look different because they express different genes. So your body has a whole bunch of genes, it's like 40,000 or so. And different cells express different subsets of those genes, and that's why one part of you looks different from another part. We know a lot of the genes that embryonic cells express, and we know what the differences are between some of these adult cells. And by expressing some of the embryonic genes in the adult cells, we can convert these adult cells back to an embryonic state.
In a very simplified example of what Dr. Thomson is talking about, we unwind the double helix of a DNA strand, revealing a ladder-like configuration. These segments are different genes, and scientists have learned how to turn them on and off. If you turn these genes on, the cell will grow into a bone marrow cell. And if you turn these genes on, the cell will grow into a blood cell or a nerve cell.
With all this news, the first question that comes to my mind is, do these new iPS cells eliminate the need for embryonic stem cell research?
I think in the future it may, but currently that's premature. It's my own personal bet that these are going to be equivalent to embryonic stem cells, but time will tell. We haven't studied them for very long. We've had embryonic stem cells for 10 years, and these are brand new. The human ones were just reported last November. And I think that they're the same, but it could be that over time we'll find out they're different in some substantially clinically relevant way. So it's good to be cautious. If they turn out to be identical in every way, I think that scientists will just kind of shift towards these new cells because there's all these difficulties in working with the old cells. I mean, it's always ethically controversial. The consent process was very difficult. So I think there'll just be a natural shift away. But for people to say, gee, we don't need it anymore, we shouldn't support it anymore, that's wrong, because it could be that I'm wrong about these new cells, and it could be that we went down a path that ended up being a dead end for reasons we can't perceive right now. We derived these newer cells a year ago as to Shinya Yamanaka, these induced pluripotent cells. And the White House and others said, see, we didn't need to do it at all. What actually happened is over the last 10 years, we learned a huge amount about embryonic stem cells. And we needed that knowledge to derive these new cells, because the basic culture conditions and the genes that are expressed, we didn't know that. So if we never had embryonic stem cells, we'd never have these new induced pluripotent cells. So science, you can't put it in little pigeonholes. It goes from one bit of science to another bit of science, and it goes back and forth all the time. And that's why when I say that my guess is these new cells are similar to embryonic stem cells, it would be a huge mistake giving out embryonic stem cell research, because I could easily be wrong, and what we learn from one directly applies to the other. And that's similar to the debate about adult stem cells versus embryonic stem cells of a couple years ago. You know, everybody keeps saying that, well, there's all these wonderful therapies on adult cells. In science, there's no distinction between the two. What we learn about embryonic stem cells directly impacts what people can do with adult stem cells, and what we learn about adult stem cells directly impacts what embryonic stem cells. So these divisions that have been created have been very much based on politics more than science.
With all this research and the amazing discoveries, where is this going? What is the purpose?
Yeah, so the purpose that I see for the embryonic stem cells and these newer cells is that they give us access to the basic building blocks of our bodies. And scientists have had only limited access to human cells previously because it's simply hard to grow them in the laboratory. So if you take a brain cell out of a dead person and put it into tissue culture, it doesn't divide. It just sits there and dies. So it's very hard to actually study that cell. We now have cells that'll make living, normal brain cells in tissue culture on demand in whatever quantities you want. And if you think about drugs that affect brain cells, well, you want to put those drugs on the target, those brain cells, and see what it does. We've never been able to do that before. We always used animal models like mice and dogs and things like that. And although those animals work pretty well, we aren't mice. We actually function rather different than mice. And studies on any animal model is limited by the fact that we're different than those animal models. So if you use actual human material to study, it should be more accurate to human medicine. It also has a side effect of reducing animal use. But that's the real value here, is it gives us access to the human body.
So if I understand this correctly, it's more about research, drug and therapy development rather than transplants.
I'm guessing that 80 or 90% has to do with basic research, and that there'll be some transplantations based on these cells that are successful, and it may well be life-saving for particular conditions. But most of the impact in medicine will be on the other 90% about the basic research. There are no human heart cell lines, so drug companies can't directly test drugs in the laboratory on human heart cells because there are no human heart cell lines. So human embryonic stem cells for the first time give unlimited supplies of normal human heart cells that drugs can be tested on. And that means that you can discover new drugs faster, but it also means they're safer because you can see if they're toxic in real human tissue for the very first time. But again, the fundamental importance of these cells to me is it gives us new unprecedented access to the building blocks of the human body.
It's been a little over 500 years since we first peered through a microscope, and over the past century, scientists have made some remarkable discoveries about how our bodies work. I hope this film has provided you with the information you need to make an educated decision about stem cell research, to help you decide where to draw your line in the sand.